In this procedure, fluorescently labeled RNA transcripts are injected into Xenopus oocytes, and their localization is monitored. First, the RNA is labeled with fluorescent nucleotides in an in vitro transcription reaction. The RNA is then loaded into a microinjection apparatus. The oocytes are placed on an injection dish and carefully microinjected. Finally, the oocytes are cultured to allow RNA localization to occur before imaging on the confocal microscope.
Hi, I'm James Gagnon from the laboratory of Dr. Kimberly Mowry at the Department of Molecular Biology, Cell Biology, and Biochemistry at Brown University. Today we're going to show you a procedure for visualizing subcellular RNA localization in Xenopus oocytes. We use this procedure in our laboratory to study the RNA architecture and protein factors required for RNA localization during early vertebrate development. So let's get started.
To begin this procedure, transcribe mRNA, which is both radioactively and fluorescently labeled as described in the accompanying written protocol. Add DNase to degrade the template DNA, and then stop the reaction by adding EDTA. Remove unincorporated nucleotides and other small molecules by spinning through a G50 column. The RNA is then concentrated by ethanol precipitation, washed, and resuspended. After determining the concentration by scintillation counting, dilute the RNA to 50 nanomolar and store in 5 microliter aliquots at -80 degrees Celsius. When ready to perform the microinjection, thaw an aliquot of the labeled RNA. Denature at 70 degrees Celsius for 5 minutes. Then spin for 10 minutes at maximum speed in a microcentrifuge at room temperature to remove particulates and keep on ice. Next, isolate the oocytes. Trim pieces of Xenopus laevis ovary into